please let me add that immune systems are found everywhere in the living world. When I started to study immunology, people insisted on the importance of the immune system in vertebrates. I think that some of the crucial changes in recent immunology is to say that you find immune systems everywhere, of course in invertebrates, of course in plants, very likely in prokaryotes. Even it's pretty sure that prokaryotes, especially archaea, for example, the CRISPR-Cas system, but not only the CRISPR-Cas systems, probably there are immune systems everywhere in nature. So I think that immune system is in fact consubstantial to life. So you cannot have a living thing without an immune system. This is what I think. And that is a system that is involved in defense, but not only defense. If you think about the immune system that way, then I think it opens up many, many different views about its role and its evolution. So this is taken from Margaret's and a colleague, a paper in PNAS that I showed before with many, many authors. And I think that they suggest something that is very important for me in terms of interdisciplinarity, which is that we should think maybe about the body systems in terms of neuroendocrino immune systems. So of course it's much easier to say it than to do it experimentally, but I think it's very important to suggest to people working in these different areas that they can interact on that kind of objects, the microbiota and its interaction with the host in general. Biological unity through biological unification. So I want to go back to my first question. Are we ecosystems? Are we communities? Or are we units? Or both? We saw that organisms are to a large extent made of microbes and by microbes. So should we say that, in fact, each of us is a plurality of entities rather than one unit, a unified entity? Is each of us a community of microorganisms and endogenous components? Is each of us an ecosystem? Some people said that recently or not so recently. Lynn Margulis, a key biologist involved in the identification of endosymbiosis with regard to mitochondria in particular, but not only, said that individuals as the countable units of population genetics do not exist. More recently, some of my friends, including Scott Gilbert, Jens Sapp, and Fred Torber, said that if you defend the symbiotic view of life, you will conclude that we are not individuals and we have never been individuals. And I disagree with that. I think it's really a misinterpretation of what biological individuality is. I think that what these people have shown or suggested is that living things indeed are complex and heterogeneous multi-species ecosystems. So it means that they are not individuals in the sense of genetically and functionally homogeneous entities. It is true that we are not individuals in that sense. We are not endogenously constructed. We are not self-constructed in any strong sense of the word. But I think that we are not any kind of ecosystems. I think we are very special. When I say we, I mean all organisms, not humans. All organisms that were described previously as having an immune system, for example. I think we are a very particular ecosystem. We are highly individualized ecosystems. And I think that the key aim, as I said, for biology and philosophy of biology is to understand the mechanisms that unify this plurality of constituents into a cohesive whole. So this is exactly what I'm calling, following the American motto, or e pluribus unum. So with several, you make one. I think that is one of the main investigations for biology. And I think the role of the immune system in this unification of a plurality process is absolutely decisive. I think the immune system plays an important role in the inclusion-exclusion mechanism. I think that the immune system says, this is going to stick together, this is not going to stick together. This I accept, this I do not accept. It's very important to emphasize that I reject completely the self-non-self view as it was suggested initially, which is a view based on the origin of the antigen. What I'm saying is that immune system constantly says, this I want, this I don't want, but they don't do that on the basis of the origin of the component that they recognize. We're full of foreign entities, and our immune systems are very happy with that. They're just great. Those bacteria are fine. And the immune system copes with them in a way that creates the kind of equilibrium I described before. So I think what we should understand is the fact that the immune system is still a system of inclusion and exclusion, but not on the basis of the origin. Do you come from the inside? Do you come from the outside? I don't care. The question is what you do as you know, as a function to the whole system. So this is what I've tried to defend, and this is why I thought that the self-non-self theory could not do, and this is also why I've tried to suggest this continuity theory, about which I'm going to say very rapidly a few words, just because I feel the need to advertise my theory. No, it's not exactly that. It's just that I think that this is one of the theories, there are others, that insist on the crucial importance of the microbiota for the functioning of the immune system. And here I'm just going to say, to do a very quick presentation of this theory, just to say that it was started in 2003, mainly in terms of research, and it was developed with Edgardo Carosella, for example, in this paper, and then developed much more extensively in this paper with my friends Eric Vivier and Sébastien Jäger, both at the CIML. Sébastien is a biophysicist, Eric is an immunologist, as you know. And we wanted to understand the role of the immune system and how the immune system works. And our suggestion is that it's a very, very simple principle. Our suggestion is that an effector immune response, in contrast with an immunoregulatory response, so I think there's no non-response of the immune system. There are different degrees of effector and immunoregulatory responses. And we think that an effector immune response is triggered by the sudden modification of the molecular patterns with which immune receptors interact. So what we say is that what is crucial for the immune system is to constantly monitor what's going on in the body, in the organism. And if things change very rapidly, then it will trigger an effector immune response. If things change slowly or don't change, then you will have immunoregulatory responses. The origin is not important. What is important is the speed of change, the rate of change, the derivative of the quantities of antigen through time. So this is why we drew this different schema, talking about the induction of an immune response according to the discontinuity theory. And here we insist on the fact that if you have the sudden appearance of an unusual antigen in an organism, you will have a strong immune response that then will be extinguished. If you have an initially unusual but persistent antigen, you will have a strong immune response, and then this immune response will tend to be extinguished. If you have the slow appearance of an unusual antigen, you will have very, very slow immune response. If you have the intermittent appearance of an antigen, in that case we have some sort of chronic, long-term, strong immune response. And we try to use that model to understand different situations, both physiologic and pathogenic. So I'm going to skip that just because we don't have much time. My suggestion, at least in my book, I don't think Eric would completely agree with that, but I think that one of my intentions in my book was to say, okay, what is true in the self-non-self theory, what is true in the danger theory, in fact, can be captured by the continuity-discontinuity theory, and not the other way around, suggesting that we can unify under this very simple explanation having to do with the rate of change. We can gather very different situations. Immune surveillance concerning cancer, the God hypothesis in plants, which is exactly an example of effector-triggered immunity, which has been known for a long, long time. And in that kind of cases, what you have is the immune system that recognizes strong modifications of host patterns, not the microbe itself, but the consequences of potentially pathogenic microbe on the host. And this is, again, the intensity of the change on the host that will trigger the immune response. I think to some extent the discontinuity theory is an extension of the God hypothesis in plants. I mean, I discovered the God hypothesis after having formulated this theory, but I think this is exactly that, just an extension of this very idea. And many other examples, I think, can be captured by this theory. What are now the therapeutic consequences of the view that organisms are ecosystems, but also strongly unified ecosystems because of the activity of their immune system? So this unification function of the immune system that I described. I need only 1 minute to finish about these therapeutic consequences. I think we're moving to a different way of seeing our interaction with the microbes. It's already something that has happened. I think it's going to continue. It's maybe not always a good idea to fight microbes. it could be a good idea to go back to this conception that sometimes what we should do is to restore body's equilibria. So I know that Gérard Hébert has important ideas about that, including in his recent theory about the equilibrium model of immunity, and I think that is very promising. Maybe in some cases what we will do is to maintain or maybe even to stimulate some symbionts. I'm very interested in the different interactions between different microbes in the body, so related to the co-infection studies that have been done in the last 20 years or so. We could even think about injecting symbiont products in some organisms. And we could also think about establishing a new gut ecosystem. And that's why I've been so much interested, as I'm sure many of you, in fecal transplantation and in some of the papers that have been published recently, so especially that one, which was much discussed in the New England Journal of Medicine, but many others were published. And as you know, it's a very cheap, it's a very nice, I don't know, it's a very interesting way of replacing the gut ecosystem of a host. There are many problems with that, many issues, ethical issues, practical issues. How long is it going to last? How does it work? Are you going to bring some infection with that? Many, many problems. But I mean, this is very interesting and very stimulating, I think, new way of thinking about our interactions with microbes. And in that case, as you know, it's a very specific case with C. difficile, which was very, very strong. So the patients were in a very special situation. But I mean, the results were so good, so striking, that they had to stop the whole thing and, and do the fecal transplantation to everyone. So I think this is a very stimulating example. Another interesting example, I think, has to do with viral therapies, the use of phages against pathogenic bacteria, and maybe oncolytic viruses, but I don't want to go into that. And I will conclude by saying that microbe-induced development has important consequences about development, about our conception of host-microbe interactions. I think that while microbes have generally been seen as what the immune system must eliminate, many microbes in fact play a key role in the construction of the immune system, both at an early stage and throughout life. I think it changes also our conception of the immune system and its role. It is involved in construction, repair, and the maintenance of the organism, and not only in defense. So please do not ask me, why do you say that defense is not important? I think defense is very important. I'm just saying that this is all that which is important. And finally, I think it has some consequences on our conception of biological individuality, and this individuality is heterogeneous, but at the same time physiologically unified, in particular through the action of the immune system. So I want to thank those people, my little group, the microbiota group in Bordeaux, Thomas Bach, John Dupré, Gérard Hébert, Scott Gilbert, Peter Godfrey Smith, Margaret McFungai, Sakis Mazmanian, because I always send them emails and very nicely they answer, which is great. And of course I have to thank the ERC because this is mandatory. Thank you very much for your attention.
So obviously now it's time for questions. My idea is I will give the microphone to the ones that are more in the front so that the ones that are in the back can listen, because I don't want to, there is only one microphone, and the ones in the back, of course, everyone will listen. So who wants to ask something? I will start with the first question then. Is there any data that can support the discontinuity theory?
I hope so.
Or is there anyone, so is Vivia or anyone working on that?
So there are different aspects. One aspect is very, bottom up, which is that we gathered some data, especially on NK cells, macrophages, just because this is the, especially NK cells on which Eric and his team work. And we have many elements that show that the different situation that I described work for some of the immune cells. Then there's a different question in your question, which is, do some people try to really assess the theory very specifically? And to that,
to challenge it.
Yeah, to challenge it and how to. So the answer is yes, but for now it's very uncertain. So there are people in Melbourne, because Eric was in Melbourne, and some people got interested. Some people in my lab, because I'm trying to sell my ideas to my colleagues, but some of them are interested in specific settings relating to CMV, and I can tell you more about that. And also some people in Germany, and also some people in Marseille. The thing is, I think what we need now is someone doing that full time. Someone willing to do that full time. Otherwise, it's many indications suggest that it could work. Indication that could go against it. So I don't think we have for now a clear answer to will it work or not on the basis of experiment that could have been done specifically for that theory. But what we did is to try to make very clear some predictions that could invalidate the theory. That would be the best way to assess it, I think.
Thank you.
Thank you, Thomas. That was great. So, have you considered the possibility, the vertebrate body plan by its nature has been thought of as being of 10 organ systems, right? And by every definition, actually, the microbiome satisfies the strict definition of an organ system, of communicating organs throughout the body. And so have you considered the possibility of thinking that of the microbiota as an organ system that it itself develops?
Yes, but then it's yes, but then it raises many difficult issues. So I think indeed this is something like that happens. I just think that if you do that, then you need to say what exactly is will count as the microbiota, what exactly will count as this microbiota-like organ, and it leads to many questions that I think are very important, which is the distribution of the microbiota in the body. So maybe you have some specific sub-organ of this microbiota. Maybe you will need to think of how these different components of the microbiota can influence almost everything in the organism. And that, for me, is extremely interesting because what I think is that the immune system is probably the only true system in the body by its influence. And what I think is that, in fact, part of the microbiota is part of this complex immune system. So what I'm trying to think about is the way this complex immune system made of different things, including microbiota, can have an influence throughout the body. So that is my intuition about its development and how it should work.
Yeah, I mean, because clearly Gary Huffnagel has shown that the different components of the immune system, respiratory and so on and so forth, communicate with one another.
Yeah, yeah. I agree.
So I'm interested in one question. So there are a number of patients that actually have, as far as we know, healthy microbiome, but then undergo surgery, extensive intestinal surgery. So they basically, their microbiome was completely screwed up. And it seems to me that, at least I can only speak of France, there is no real interest in fecal transplant or auto-transplant, because I realize that the problem of fecal transplant is always also, are you transplanting an organ? This is a self-replicating entity, right? So I'm curious about that. I'm curious about whether you have thoughts about that, because this is a...
So you mean specifically about why France is not?
No. About the question whether this is a person whose microbiome is changing due to external influence. Is this a changed entity? Is it the same person?
Well, it's a very good question. It's a very good question. The answer is I don't know, and I'm very interested again in the rate of change. In what you describe, it's very rapid. It's interesting to see what the consequence of this rapid change, and also in the reconstitution. What's going on after that? I don't know about the specific case you're describing, and I think it would be very interesting to go deeper into that. What I know is that some people thought about changing the microbiota of mice, for example. And what I heard is that it's very difficult to do because there are all sorts of niches. So Masmanian worked on that, for example, and realized that it can be recolonized. So I'm sorry to be so vague about that because many of you are specialists, but it seems that there is, for some time, a change in this composition, and then it goes back to something that that it was before. So I'm very interested in knowing more about that. So in which cases the community will change for long, in which cases it will go back to the previous state, in which cases the change will be so quick, so fast that, for example, there will be no new adaptation of the system, so it will be completely new. That is exactly the kind of questions that are, for me, fascinating. But of course, there I need to know the data, and I don't. You know, so these different change-related questions are exactly the things I want to investigate.
I think there's a paper on that showing that there is a change with
Okay, so
big exposure to aerobic environment for several hours. I think there's a paper on that. I wouldn't be able to cite it.
okay, okay, but I would, if you could send it to me, and maybe I don't know about that. I just think that probably it would be very interesting to confront that to, you know, different papers. So like in the fecal transplantation literature, some people said you can change completely the composition, and then others said no, it's not going to be the case on the long run. So this in the long run. So this really, and I don't think that many people have, you know, monitored people for very long, etc. I think we lack the data about that kind of examples.
Very nice talk, Thomas. Thank you very much. As you know, I have an issue with individuality, and my opinion is that, not for myself, well, maybe a little bit.
We can talk about
that. So my opinion is that individuality is scalar, that you have, you know, scalar symmetry at the cell level, or, you know, a group of cells, a human, a society of human. But that's, and if you use the immune system as a way to define the individuality, it's probably also problematic, because the immune system is scalar as well. You have, you know, immune system in the cell itself. You know, as you mentioned CRISPR-Cas, for example. That would be one immune system at the bacterial cell, but that's also true for, of course, for the mammalian cell in terms of type 1 interference. And by itself, a cell could be defined as an individual because it has a system in a cell to defend itself. And then you can take this as kind of common sensing between different cells, because if a cell is infected or is stressed, then it signals to the other cell to do the same.
But you exactly confirm my point of view is everything in biology is community-like. Then there are degrees of individuality. Those degrees of individuality are related to some mechanisms of unification. There are many of them. One of them is the immune system. The immune system can be in a cell, it can be in a multicellular organism, it can also be social immunity. I've taken in my book examples of insects, where it's very clear that the social immunity is in some cases stronger than the individual community, as we would normally. So I'm not talking about common sense individuals. I'm saying we have the science to show that some communities are more individualized than others. And this is what I'm interested in. And I took many examples of colonial organisms, Botrytis laceri, and examples of corals, in which you can make a difference between different cases where the immune system does not play the same role in different circumstances. So you can say that in some cases, it is more like an individual than in other cases. But having nested individuals is perfectly normal in the literature. So if you have a look at the literature about evolutionary individuality, it's just about nested individuality. You have individuals within individuals within individuals. The key question is to avoid the view that, oh yeah, you know, we don't know. I think that is not a good view. The good view is to say we have criteria for defining individuality, and these criteria are multiple, they are diverse, and they should be articulated to end up with something precise. And I think that is important, because it has some functional consequences. If you study something that has an immune system, is unified by an immune system, I think you can find other things than if you study a system, for example, plurality of cells that does not have this immune system. So this is my point. My point is individuality is related, but it doesn't mean that everything goes. You know, there are some degrees and some processes and mechanisms that will instantiate this individuality thing. That's my point.
I think he was no?
Sorry.
Ah, no. He was asking for the... okay.
First of all, thank you for your talk. And I have a more philosophical question. Throughout the history of philosophy, soul has been defined as the unifying principle of all the different activities of human body. So, in a very provocatory way. Do you think that one day immunology could fulfill the crazy dream of 18th and 19th century materialist philosophers and finally prove that human soul is nothing but a product of our guts?
That's a very provocative question, a very good question. So, part of the enthusiasm that I described, so why are philosophers so enthusiastic about the brain-gut axis, and it's related to what you say. We're interested in what creates a unit, and we're interested very often in what is thought. And thought can be sometimes conceived of as a soul-like thing. So some people don't believe in souls, as you said, others do. I don't think that the immunology will show anything like that, but still, it is very clear that you can see now in philosophy people going to the science, like me, to find partial answers, and you can see the resistance of other people. So we can see, so where I was in the philosophy department at the Sorbonne, you can see that. Some people hate the science. They don't want to know about the science. And it's very clear that they feel threatened by the science, or at least they don't want to hear about it. And there are other people who, I think, maintain a long-term tradition in philosophy, which is to say, if we demonstrate that these bacteria have an influence on our thought, it's interesting, and let's see how it goes. So, of course, we're not going to demonstrate what you were talking about, but still, I think some people are really feeling the need to answer to that or say science cannot say anything interesting about that. And the neurosciences, of course, in relation to the kind of material I described, are very important there. And it's very clear that if you talk to a philosopher, especially in France, in Germany, they hate neuroscience. Many of them hate neurosciences because the neurosciences are going at the heart of philosophical questions. And you too, immunologist, and you do the same. Maybe it's less clear, but you do the same. And my aim is exactly to say, well, fine, let's see what these people say. And we philosophers still have a role in putting that together, raising questions. Some people, so sorry to dramatize a little bit my answer because you were making a joke, but an interesting joke. And I think that is really something going on now, really this tension between scientifically oriented philosophers and less scientifically oriented philosophers. In France, it's something like 3%, 97%. In other countries, it's very different, and of course, it's nicer for us to go to other countries where it's more possible to do that kind of thing. But you see, I think this is a very important question, and we'll see in the future how more traditional philosophers react to that kind of literature, gut-brain axis, if it's confirmed, if it's stronger. I think there will be some reaction.
I understand from what you said that the immune system is involved in genesis of organs. And I am working in an organ which is called the brain, and we hear about the immune privilege of the brain. As far as I understand, it means that the antibodies, for example, and the immune cells cannot penetrate through the blood-brain barrier during crucial steps in the development of the brain. How can you conceptualize that? So I have a hypothesis, but how do you?
So it's a crucial question here again. So what I think is that when I started immunology, there were many immunoprivileged organs. Now, not so many. So the first thing I want to say is that we'll see. That's my first answer. Then there are two aspects in your question. One is related to, is there some microbiota in the brain, and one is there some immune system in the brain? And these questions are related according to what I said, but they're different. So what I think is that it is very clear that there are immune elements in the brain. The question is which one? When do they come here? What do they do exactly? So the examples about the microglia, I think, are now very well discussed. It is also clear that not any immune entity goes to the brain at any moment. So I don't know about this dynamic. So maybe you're interested and you can tell me more about this, when they arrive, what they do, etc. What I want to say is that there is an immune system in the central nervous system. There is an immune system in the brain. This is very clear to me. It is not clear that there are microbes there. Maybe you know more about that. But what I think and I just gave some examples of that, is that the microbiota can influence an organ without being in that organ. You can perfectly have some strong influence on the brain by the microbiota of the gut, for example. And I think that is crucial. We don't necessarily need to demonstrate the presence of X to demonstrate an influence of X in a given organ. I think that is crucial. So that would be my two answers. But maybe, I'm not, here again, I want to know more. You know, if you study this developmental aspect, I would be very interested.
I have a hypothesis. I'm working on cell adhesion molecules, which are very on the surface of neurons and glia, which are very rich in immunoglobulin domains and very complex, and with a lot of combinatorial states. And protecting the brain during development of the tissue, the neural tissues, against the penetration of antibodies, for example. This is a way to protect from interference between normal and pathological, reactive immunoglobulins, which would, if the blood-brain barrier was permeable to those antibodies, would screw up completely the development of the neural circuits. So it's selective, the results of the selection.
Sure.
This is an hypothesis. How do you?
Well, and then these molecules disappear. So then these protective molecules, what happens to those molecules?
Not protective. Cell adhesion molecules, which are involved in the specification during development of neuroblasts, neurons, then neurites, and then synapses. And there are many myriad of molecules bearing immunoglobulin domains are involved in this. And if antibodies coming from outside the brain, from the immune system of the body, would enter into this developing neural tissue, it would screw up completely which happens in pathological situations.
Okay.
So my hypothesis is that the immune system might not be, hopefully, involved in development of the brain. I mean, immunoglobulins from outside the brain.
Okay. Okay.
So this would be an exception to your hypothesis.
Possibly, but it's about.
You can discuss.
Okay, yeah, we can discuss about that. Yeah.
Okay, it seems time to close because people are already escaping from the room. I wanted to ask you something else, but I will just leave it out as a comment. I really like your idea of redefining the immune system, not only as a defense system, like also repair, construction of the body. And here it fits a bit what there is a hypothesis as the immune system as a cognitive system that was developed by Antonio Coutinho and Francisco Varela. We can think about it, but that also gives me the idea that actually all systems could be redefined depending on the all other functions like you could find, not only the immune system, right?
Can you say that again?
That because you are saying that the immune system has more functions than defense, and this idea could be extrapolated to other systems. That's just a comment, and
I agree.
Everyone would agree, I guess. Okay, with this, sorry, but I think it's has been a very nice talk and seminar. We have discussed a lot. Also, thanks to the people from the immunology department for co-hosting and for distributing for advertising the seminar. Maybe some immunologists wants to go to study some plant development, as Thomas is saying, proposing. I just want to say the next seminars from our series. The next one will be on the 27th of June, and will be given by Antonio Lascano, who is an expert, is a